Okay, well, welcome. This is the last meeting of E340, 542, Dynamics on Networks. Today we have the final presentations from our three students. As always, I remind you that this is recorded. If you don't want your material to be made available, please text me and we'll cut that out of the recording when it's distributed. So on our agenda today, we have the final project presentations, and we'll start with Roying. Is that okay?
Okay. So the project is about modeling the MAPK and AKT signaling and the drug inhibition to this signaling pathway. And the idea is coming from the paper called the computational model on the modulation of mitogen-activated protein kinase and AKT pathway in Herguelin-induced ERBB signaling. This is a paper from 2003. The paper studied how a ligand called Herguelin activates the receptor ERBB4, which triggers two major pathways. One is the MAPK to ERK, which control the cell growth, and the PI3K to AKT, which control the cell survival. And why this is important? There's in the cancer cell, many tumor has high ERK and AKT level, which will result in uncontrolled cell growth and survival resistance. So it will keep growing and survive. So if we can find a good drug target that inhibit both ERK and AKT, then we can probably control the cancer cell growth and survival. And since there is a cross talk between these two pathways, so it may cause drug resistance in some cancer. And the goal is to identify a good drug target for cancer treatment. And we want to find out how do different molecular steps contribute to the ERK and AKT activation, and which reaction are most fragile or influential, which is a better target. And we want to find which pathway nodes make good drug target. And what happened when we combine two drugs? And there is some challenge because some reaction use Michaelis-Menten forms, and we cannot modify the Michaelis constant. And there is and we need to interpret it which species corresponded to the druggable proteins. And this is an overview of the the model. So the Herguelin coming as the input, and it is a ligand which will bind with the ERBB4 and result in this RP. And RP will trigger two pathways. One is the MAPK pathway through this to here, and the other one is the AKT pathway that will result in AKT double phosphorylation. And this MAPK pathway is a little bit more complicated than this AKT pathway because the RP does not directly control the production of ERK double phosphorylation. It controls through catalytic reaction. So it makes it more complicated. And this RAS is already simplified. This cycle is simplified. And there's two types of reaction in this model. One is the mass action, which just directly generate this species through this reaction. And there's another one. There's another reaction type, which is the Michaelis enzyme, which will generate which will catalyze the reaction, and it did not involve in the reaction directly. And this model assumption is it's a ODE model. It does not have stochastic. And the author assumed the whole cell behaves like one well-mixed compartment, and everything interacts instantly in the same volume. And they record the system in a short amount of time, which is 30 minutes. And they simplify the RAS cycle. And for the sensitivity test, I assume that drug inhibit through scaling the rate parameters. And in the drug test, I treat drug as species binding inhibitors, not directly rate cancellation. And I only focus on the peak activity instead of the full time. And I try to reproduce their result. And as we can see, they're slightly different due to the typo in their model. So they write AKT PIPP here, which should be AKT PIP. So this typo causes slightly different peak level of AKT. So in this reproduction, we have slightly lower AKT level. Where's AKT? No, ERK level. Slightly lower ERK level and almost similar AKT level. And I also reproducing the drug effect, drug sensitivity test they did. I will show you in the notebook later. And I also did 1 and 2 factor sensitivity test, which will find which species is a good drug target. And in this sensitivity test, I find that K11 has the highest influence, and SHC, which actually can control both ERK and AKT, which is unexpected. And for the drug inhibition test, I simulate the drug effect by binding a drug to the species, and this binded species won't take part in the reaction. So basically, it's just taking amount of the species away from the reaction. So just reducing the species. And I'm setting the K on and K off using the IC50 value from the GDSC. So this is a drug that inhibits PI3K, and its mean IC50 value is 3.15 millimeters. And the definition of IC50 is the concentration of a drug or a compound needed to inhibit a specific biological process, like cell growth. And AKT controls cell survival. And here, when the drug concentration is 3.15, it should be hit the 50%. So that's how I tune the ratio of K on and K off. And I also did a two drug combination test, which shows that a combination of drug can actually be more effective than using one drug alone. So as we can see here, so if we use the more effective drug, like the drug one here, so if we use 10 millimeters, the ERK-PP level will reduce to 18%. But if we use 3.15 and 3.15 drug 2, we will actually reduce more peak level. And we also find that the drug simulation test is not equal to the parameter sensitivity test. So we find that K11 is a very effective parameters to tune. But in fact, when we try to bind the drug to the corresponding species, it's not as effective as expected. And let's take a look at the notebook, which will better explain what I just showed. So first, I reproduce the image. So basically, it's just the different amount of ligand. So I just changed different amount of input value of the Hergulen, which is the ligand that bind with ERBB4. So a higher amount of Hergulen will activate the whole system more. And it's a little different from the one from the paper because of the type. And I try to reproduce the figure 6. So they are trying to do the drug sensitivity test too. So they said that Wortman is a fungal metabolite that can inhibit PI3K. So they scale the K19, which control the PI3K level, and see how will it affect the ERK-PP and AKT-PP peak. And since PI3K only affect the AKT-PP reaction, so the AKT-PP will drop when they reduce the K19 value. And since AKT-PP will inhibit ERK, so without the inhibition of AKT-PP, ERK-PP level will actually increase. So here's an exercise. So if we add one more control, which control the K19. Oh, this one is K27, sorry. So if we add a K19 parameter scaling, and how will it affect the AKT-PP level and the ERK-PP level? So K19 is here. So from ERK to ERK-PP. How will it affect the AKT-PP and ERK-PP level? So we can slide the slider here. You can slide the slider here and see how will it affect both peak level. As we can see, the answer is it will reducing the K19, it will only affect the ERK-PP level, but won't affect the AKT-PP level because we can see that K19 will only control this pathway. So it won't affect this one here. So did the sensitivity analysis to identify promising drug targets. So the goal is to find which part of the ERBB4 signaling network have the strongest control over the ERK and AKT activation. So I did a one-factor sensitivity analysis and reduced the parameters to 90% of its original value. And I measure the ERK-PP and AKT-PP level. And the reason why I choose the peak level is that these models are transient. They rise to maximum and then decay back to zero due to the feedback and the dephosphorylation reaction. So we only need to care about the peak level. And for the forward reaction, I did a sensitivity test and find that the K5 is the most effective parameters. So K5 control the amount of RSHC. So reducing RSHC will reduce ERK and AKT peak. And here's the conclusion of the one-factor sensitivity test, which shows K5, K6, K3, and K1 and K2 is more effective to the ERK, and K23, K24, K25, K29 is more effective to the AKT peak. So here's an exercise. Why SHC will affect both AKT PP and ERK PP? Since SHC only controls the RSHC and it should only control the pathway in the left side, which is the ERK pathway. It shouldn't have any control to the AKT-PP level. But in the sensitivity test, when we reduce the RSHC, it will actually reduce AKT as well. Should we do the exercise here now?
Yeah, please, that would be great.
And here's a hint. It's because of one of the reaction can actually affect the AKT level. And here's a plot that will explain it.
Why, do you want to walk us through that a little bit? I'm not sure I could understand it just from the diagram.
Okay, so why reducing the amount of SHC will affect both pathway? So this SHC, after reducing the SHC, this RSHC will decrease and this will decrease. And since the the enzyme of this reaction will decrease, it's reduced. So this reaction will reduce. So there will be less RSGTP, less RAF star. And so this activation of ERKPP should be less. But why it will affect the AKT? Because SHC is not in this pathway. It doesn't directly control the AKT-PP level. But in the sensitivity test, we see that SHC can control both ERK and AKT-PP level. So it's very confusing. So why SHC will control both pathway?
Yeah,
So
because you have less RP on the pathway on the left, which means you have more RP on the reaction on the pathway to the right.
That's our original thought, but
Yeah.
I plotted here. It doesn't seem that way. So RPI level is actually higher. So there is more RP in this reaction, in this system. So this is not what we assumed. So the answer is in this reaction. So AKT double phosphorylation will goes back to the AKT PIP3 when this MEKP level is lower. So when this MEKP level is lower, then this back reaction will be faster. And this will cause the AKT dephosphorylated very fast. So this AKT PP level will actually decrease due to the decreasing of MEKPP.
Oh, so are you saying that so basically the diagram is wrong because the diagram doesn't show backward arrows,
There is
right?
a back, but it doesn't show MEKPP as the enzyme
Uh, wait,
of
wait,
this
wait.
reaction. So there is backward. The
Ah,
react,
yeah, okay.
so it does not show this one is the enzyme of this. So it's very confusing.
Oh, okay. So it's incomplete.
Yeah. It's
Mhm.
yeah, it takes me a lot of time to find out why this SHC can actually control AKT, because through the diagram we cannot see why it can control the AKT. It doesn't make sense since I plot the RP level, which is actually increasing, which is unexpected. And so the answer is that it reduced the MEKPP, which is the enzyme of the dephosphorylation of the AKT-PP back to the AKT-PIP3. So this is very confusing. And this is why scaling of K21 won't affect the AKT, because K21 is here. So it directly reduce the amount of ERK double phosphorylation by inhibiting this reaction, and it won't affect the MEKPP level, so it won't affect AKT. So in the sensitivity test, we will see K11 is very effective on both pathway, because this is the enzyme of the RAF star reaction, and RAF star will control the MEKPP level. So this controls this RAF star, and this controls this MEKPP. So reducing the RS-GTP will actually affect both pathway very effectively. That's why K11 is the most effective parameters in this whole system. And we found that K11 is very effective. And for the two-factor sensitivity analysis, so I want to find if combination of two drugs is actually more effective than using one drug only. So basically, it's just the one-factor sensitivity test using two drugs at the same time. So the result is very expected because K11 is very effective. So everything in this two-factor test will have K11 due to that it's very effective. So since it's too influential, so I removed the K11 from the two-factor sensitivity test to see if there is another effective drug combination. So we can see here that K5 and K8 is the only drug combination will reduce ERK and AKT peak level at the same time with very effectively. And the other drug will only affect one. So I will do the drug combination test later. And to validate that this potential good drug target from the sensitivity test, so I tried to simulate the real world scenario. So I will bind this drug to the species directly instead of controlling the reaction parameters directly. So this will simulate how most drug will affect the cancer cell. So drug will bind with some species and reduce the amount of protein in that cell. And I simulate this by binding a drug to a species and reduce the amount of that species from the reaction. And so there will be lower amount of that species in the system, which will inhibit the downstream. And here's the result of the drug binding test. So this SHC is corresponding to the K5, which is very effective as expected. And reducing the AKT will, this is also expected because directly taking out AKT will reduce the AKT-PP double phosphorylation. This is expected. Since there is less AKT, there will be more ERK due to the inhibition, the crosstalk between these two pathways. And this is very unexpected because RAS, which is the K11, which correspond to the K11. So K11 shows to is the most effective parameters when we do the sensitivity test. So when we bind the RAS GDP, it should be very effective as this, or more effective than this target. So it's actually not effective at all, which is unexpected. So that's why we need drug binding test instead of just doing the sensitivity analysis, which control the rate of reaction directly. Because in reality, sometimes it's not the same as the sensitivity test that can change the rate reaction directly. Sometimes it's using drug to bind with the species and see how reducing the species will affect the system. And I also try to bind with the RAS GTP directly, which is more effective than binding the RAS GTP. And this is the test they did. This is simulating the drug that target PI3K star, which they did the similar test, but they just modifying the K21 directly. No, this is K19. They modify the K19 directly to see how will PI3K star affect the whole system. And here we bind the drug with the PIK3 star, which reduce the amount of PIK3 star, and see how it will affect the system, and it work expectedly. And so most drug is acting expectedly, and it's agree with the result from the sensitivity test. But RAS it does not agree with the sensitivity test due to the fact that there is an initial value of RAS GDP. So if the drug binding reaction rate isn't fast enough, there is still a large amount of RAS GDP generated, and it will serve as the enzyme as the downstream reaction. So if the drug binding rate is not fast enough, then the downstream reaction is unaffected. And there's another exercise here. So we try to bind with the PI3K star, but how will it affect when we bind with the PI3K directly? So in the paper, they try to modify this PI3K star directly, but how will it affect the system if we bind the drug with the PI3K directly? Will it be more effective or less effective? So this is the exercise here.
Can you repeat that, please? Let's see. The question is,
So here is the result that it bind with the PI3K star directly.
Right.
But people care about the PI3K star more, or PI3K more, because PI3K star is just a species so it's not a real species. There is no PI3K star. It's just the active form of the PI3K. So people may care about the PI3K more. So how will it affect the system if we bind the drug with the PI3K directly? Will it be more effective or less effective than the PI3K star? So here is the PI3K star here. So we can think of the binding as reducing the amount of PI3K star or the PI3K. So reducing PI3K star or reducing the PI3K will be more effective controlling the AKT level.
I would have thought reducing PI3K would be more effective, but if I'm wrong, I'd like to understand why.
Oh yeah, it's more effective. So for the same amount, it will reduce the peak level more, but it's very similar to the PI3K star. So basically, they are very similar, but this binding with PI3K will be slightly more effective. And here's the reason why targeting RAS won't work as expected. So if we bind with the RAS GDP, the RAS GDP level isn't decreasing as expected. So basically, it didn't change much. And the RAF level, which is the next reaction controlled by the RAS GDP, isn't reducing as much. So that's why binding with the RAS GDP isn't as effective as in the sensitivity test. So here's the drug combination test. So K5 and K8 is the most effective drug in the sensitivity test. So let's see if it is also very effective when doing the drug simulated test. And yeah, it's also very effective. So binding with the SHC and the SHGS is very effective. And it's actually more effective than using one drug alone. So basically using 3.15 of each drug at the same time will reduce the peak level more than using 10 millimeters of either one of the drug. So drug combination is more effective than using one drug only. And that's the conclusion. And I think that's all.
Great. It's a lot of work. I mean, you asked if a report would be useful. I understand you've done a huge amount of work here, but I think this is a case where a written report might actually be useful, because there's so much material here. And I think it's not so easy perhaps to understand it in detail from the notebook on your own. But I also understand that you have a finite amount of time. I did have one comment which I should have gone back in, which I should probably have discussed or could have discussed in class, which is when you show, could you show the two-factor analysis, sensitivity analysis? Where you have the heat map.
Heat map?
The heat map, where you show the two-factor plot where you have those there. Yes.
This isn't the
Right. So maybe trying to look in more detail. So typically what one does when you do these two-factor analyses like this would be to look at the difference between the expected value if you have a linear combination of the two doses separately. In other words, suppose that you had 0.1 of dose 2 and 0.1 of dose 1. Then you might expect that you have 17.7 plus 86. And so then the linear combination would be either the sum or the mean of those, depending on what kind of an interaction you expect. And then the question is the value when you combine them more or less than what you would expect if you had them acting independently of each other? And then the places where the effect is not only bigger, but bigger than you'd expect for the combination become interesting. Does that make sense? So you basically would subtract out the background linear combination between the two.
So this should be linear. So this.
No. So what I'm saying is the following. So look at the row, the leftmost column.
This column.
That's the effect of drug 1 by itself, right?
Yeah.
Right? That's the effect of drug 1 on its own.
Yeah.
And now look at the top row.
Yeah.
That's the effect of drug 2 by itself.
Yeah.
So if I were in the middle, and I might expect that what I get is, say, am I adding the drugs, or am I using 50% of each drug when I go to the middle? I'm adding the drugs, right?
Yeah.
So you might expect that the change would be what in the top row, it's 91%, right? So there's a decrease of 9%. In the left-hand column, it's 86, so there's a decrease of 14. So you might expect a decrease of 23 if they just acted independently of each other. And the value is 77. So basically, that's exactly what you'd get if the drugs behaved independently of each other. And that may be fine. That may be all you want. But what's often interesting is what happens if the drugs aren't just acting independently of each other, but are actually interacting, in which case you'd get a number that's either more or less than the sum of those two numbers. So we could take a, let's look at, so that's what I was going to suggest. You might think about that.
Okay. So this two drug is working independent actually. Yeah.
In that case, they seem to be working independently of each other. But there might be other regions where they work synergistically or where they oppose each other. So it's interesting to know, do the drugs actually reinforce each other? Are they independent of each other, or do they oppose each other? And so that's something that it's a little bit tricky because it's not quite as simple as simply adding the effects. But the linear mixture model would tell you that. And I'm certainly not expecting you to do that here, but if one wants to do these two-factor analyses, that's something that one might want to think about.
Okay. So, but this shows that these two drugs is opposing each other, right? This one is 29, this one is 24. So it should be over 50. But
Right.
30.
Right, although you have to be careful because it may be whether you expect them to add, you have to look a little bit about whether you expect them to be multiplicative, to additive, because the percent of baseline may not be something where you expect things to add. If we look at right, you have a log scale on the x-axis. You have a log scale here on dose.
Yeah.
And the efficacy basically looks like it's almost going like the logarithm of the dose, right? The efficacy is changing almost linearly as you multiplying the dose. So it may not, so a pure sum may not be the best way to do the background. So what we'd have to think a little bit more about what the appropriate control would be. But at least to begin with, looking at the sum is not a bad thing to do, at least for low doses. For very high doses, it wouldn't necessarily make sense. You might wind up, for example, getting a negative. The percent of baseline might go negative, which you can't do. So you might have to look at fold changes of activity rather than reduction in percentage. But it's worth thinking about that a little bit. I'm not expecting you to do it here, but I wanted to mention it as something that comes up when you're doing two-factor sensitivity analysis, which we didn't have a chance to discuss in the class. Are there any other comments or questions for Roying? Again, we had plenty of time to have discussions last time, but of course, people are free to make comments and ask questions now.
I put my comments in the chat. So, yeah, I mean, my general impression is positive, so I don't think I have much to. My only issue is there is a slide where rowing implies that Michaelis Menten functions pose a challenge, and that was weird to me.
Well, because it we shouldn't change the Michaelis constant directly.
Right. Right. You can't change the half maximal concentration. That's true. You could change the rates or the concentrations, but not the but the reference concentration is something that's fixed by the physics or the chemistry and not something that you have a control over.
Yeah, that's why I only changed the
Right.
rate of the catalytic.
Right. Right. Well, I think you're using the drug to do sequestration is good is a nice way of studying that. All right. Well, thank you very much. Appreciate that.
Thank you.
So next up we have Lakshmi. So I will let you take the screen share and look forward to hearing your presentation.
I better share my entire screen so that I can use both presentation and the code. Okay, I hope this is visible. I'll start.
Go ahead, please.
Yeah. So with this presentation, I'm going to be presenting the work that I've done with modeling approach. The main topic that I've chosen is metabolism in cancer cells. Should I start my video? I think my camera is not working, so I may not be able to do that.
If you don't have a camera, then you don't have video. Sorry about that. Start
Yeah.
over, please.
Yeah.
It's just if you're recording it, it's nice to have the video, but it's not necessary. Sorry, I didn't mean to interrupt you. I should have said something at the beginning.
Okay.
So
Sorry.
please start from the beginning, and we'll get a clean recording of this. Thank you.
Of course. Yeah. So my project title is Metabolism in Cancer Cells. So I have tried a modeling approach to find out the metabolic pathway in the cancer cell. Yeah. So the main questions that I'm going to ask in this project is how the dynamics of glucose and lactate nutrition is being consumed by the cell, and how the metabolism, which is the anaerobic and oxidative respiration, is going to take place with the tumor cells which are cultivating in vitro. And also, I have introduced the oxygen dynamics to the model. So to view whether the aerobic respiration or anaerobic respiration is working accordingly with the oxygen dynamics in the medium. So mainly this will also help us to track how the cell is proliferating and how the cell responds if there is a lack of oxygen in the system. This also helps the model to add the necrosis of the cell to track that. And why this kind of study is important. So modeling such metabolic pathway will actually help us to study the tumor behavior in varying environmental conditions. And it also help us to just see the novel combination of therapies in such cancer uh, environments in vitro. Yeah. So, starting on with, these are the three key metabolism which we are going to take into a look. So basically, when a cell is growing in a medium, it need energy, and that energy is provided by ATP. In order to generate ATP for an energy, the cell will undergo such metabolisms. Either it will undergo aerobic metabolism, which we call it as an oxidative phosphorylation, or anaerobic respiration, which is a glycolysis. So in oxidative phosphorylation, cells usually consume glucose or lactate and they produce ATP. And with glycolysis, which is anaerobic, this will happen when there is no oxygen and cell will use glucose uh, and it will result in lactate and ATP. So sometimes this anaerobic respiration is we can find it in a normal cell types or abnormal cell types like a cancer. So these are the major respiration or metabolisms that will happen. So how are we going to program ourselves with the characteristics of the cancer? So when we see the tumor cells will follow this Warburg effect, which is nothing but even though there is going to be an ample amount of oxygen present in the medium or in the plasma, tumor cells will undergo glycolysis, which is anaerobic respiration instead of aerobic respiration. The main reason being the anaerobic respiration is much faster, and even though the number of ATPs which is going to be generated by these tumor cells are going to be less, but the respiration rate will be very higher, which will help the tumor cells to proliferate faster than the normal cells. So that is exactly why these effect is being seen more prominently in cancer cells. So having said that condition, so we are going to like imagine that we are growing a cell in a petri dish, and we want these cells to be the tumor cells because we are trying to analyze the metabolism of cancer cell. So our cells are programmed in such a way that they will follow this Warburg effect, which means they will undergo glycolysis in default. So when they do that, so we have a cell population. So PG is the population of cell in a medium which undergoes glycolysis. So basically, this glycolysis cell, they consume glucose and they will produce lactate and ATP. So while this particular reaction is taking place and cells started to proliferate, this resultant product, byproduct lactate in the medium will start to increase. This lactate will increase the acidity of the medium. So when the medium is going to be acidic, every cell will have a certain sensitivity towards this acidicity in the medium. They will switch from glycolysis to oxidative metabolism because they have sensed that the pH is being reduced because of the lactate increase. So when the cell is switched to oxidative metabolism, here with oxidative metabolism, we have two different types of consumption that can happen. Either the cell could consume either glucose or lactate in order to perform the aerobic respiration. But what we want the cell to do is consume the lactate because this will help the cancer cells to reduce the plasma acidity and it will bring out the balance so that they can continue proliferating like how the cancer cells usually proliferate. So in order to do that, we are introducing a parameter to our system called um, MCT transporters. These transporters will allow the cell to be more permeable to the lactate instead of glucose. So we have two different transporters. One is MCT1 and MCT2. So this MCT1 transporters will allow the cells to be more permeable towards the lactate, which will make them to consume more lactate compared to the glucose from the medium. So which will helps the cell to balance the pH of the medium while they switch from glycolysis to oxidative. Once the medium has become to normal, again they will switch back to glycolysis when we provide more glucose to the medium. So having said that, so this is how the system looked when we observe these metabolisms individually in the medium. So this one is our system. Our cells are only capable of undergoing the glycolysis metabolism. So we can see the left plot is the substrate concentration variation. So the G is the glucose concentration and the L is the lactate concentration. And the plot in the right side is the proliferation of the cell, how they are taking place. And here we are analyzing only with glucose. So in this closed system with only glycolysis metabolism, we can see the glucose started to consumed by the cell, and the cells are starting to proliferate. And by the time the glucose is started, the byproduct of lactate started to increase. And when the lactate is starting to increase, the acidity of the medium started increasing, and the cells have sensitized this acidity in the medium and they have stopped proliferating. And here, with the system only with oxidative metabolism, we can see we have both lactate and glucose in a medium, but we have made the system to behave in such a way it consumes lactate first, because we have we have set the MCT transporters so that the cells are more permeable to the lactate. So once all the lactate has been consumed, then the remaining glucose started to consume, and we can see the cell proliferating in the medium. So these are the systems which is being individually analyzed when the cell has only the glycolysis medium and the cell has only the oxidative metabolism. Next, so we were talking about the switch between the glycolysis and oxidative metabolism. And this metabolism we are introducing to the system through switches. So the first switch that we are going to see is XL. So this switch is made in such a way so here we have to see two conditions. So one is the sensitivity of the cell towards the acidity, so which is decided by the gamma. So this parameter, we are keeping it constant throughout because we are trying to recreate an ideal system for our model here. So we kept the sensitivity as 100. So which means if there is going to be the change in the lactate, the cell is 100% is going to sense that particular shift. Later, we have one more parameter called L star. This is the ability or the sensitivity of the cell towards the lactate threshold. So which means if my lactate is going below L star, my cells will not be sensitive enough to consume these lactates and produce cell will not be sensitive enough to consume this lactate. So giving this tan H function of this particular value, the resultant will be the increasing function, which means if my L is going to be equal to the or lesser than the threshold of this L star, then this switch will be off. And if my L is going to be greater than the L star, which means the level of lactate in the medium is bigger, higher than the threshold that we have set to the system, I will have 1 as an output, which means the switch is on. So it will act like a switch, which will have the output only when the lactate concentration in the medium is higher than the set threshold. And next is the switch for G. So this will act in such a way when, so the same thing, when my glucose concentration in the medium is going to be greater than my minimum threshold, it is going to be on, or if it is going to be off. So what these switches helps is, these switches will help in our equation in order to switch from glycolysis to oxidative and switch back from oxidative to glycolysis. And these are the indicators that we are using in our system. So having used that, now we are going to combine both the glycolysis and oxidation in our medium together. So now this is the medium that we have designed, which will have the both glucose and lactate consuming, and it will show this particular response. So if you see this both plots, so initially we have the glucose concentration of about 7, and we have a very low to zero lactate concentration in the medium. And we started with the initial cell population of 0.11 with respect to both cells which are undergoing oxidative metabolism and glycolytic metabolism. So initially when you see, since we are having a cancer cell and it is undergoing Warburg effect and it will undergo glycolysis first, it starts consuming glucose molecule and started producing the lactate concentration. At the same time, it is the proliferation due to glycolysis has been increased, while the oxidative response is really silent, which means like all the cells which are initially was undergoing oxidative response suddenly shifted towards the glycolytic responses. And when we see the lactate concentration is increased maximum and the acidity of the medium has been detected, then the cell started switching from glycolytic to oxidative response, and the proliferation due to oxidation has been increased in parallel to, and we can see the lactate from the higher value, it started consuming and it started uh, reducing at the end. So this is the system with both glycolysis and oxidation together. And as we said, now we are introducing oxygen dynamics into the model to see whether it is agreeing in accordance with the oxidative proliferation with the system. So in order to do that, I purely made the system. I mean, like I introduced the oxygen switches parallel to the oxidative proliferation, and the consumption of oxygen would be at the rate of um, 3 molecule per lactate, because that is how the one mole of lactate will consume 3 molecule of oxygen. So that's how I kept it done. And we can see initially the lactate started consumed by the medium in the cell, and we have like the proliferation purely oxidative, and with respect to that the oxygen is uh, reducing. So I will shift to the code before I discuss the exercise. Yeah. So So these are the documentation. Yeah. Let's take a look about how the oxidative proliferation equation has been set. So, so this is the rate of change of cell population uh, which undergoes oxidative metabolism. So the first term here is nothing but a standard logistic cell proliferation in a medium. So when uh, this is like the standard increase of the cell population with respect to the rate. And here the population, the total population, we assumed it with the percentage. So the maximum of 1, and then we have done. And the second term is the one which governs with lactate switch, and which takes care of the cell switching from glycolysis to oxidative. And the third term will take care of the cell switching from oxidative back to glycolysis, and cell proliferation due to glycolytic cell, like the same equation complement to what we have seen with the oxidative. Um, and the glucose consumption is being given here. So here, the first term is the consumption of glucose due to oxidative respiration. So this one, this is what I was talking about, the MCT transporters MCT1 and MCT2, which is governed by the parameter lambda. So we have set the condition in such a way like the cell is more adhered to the lactate than the glucose. And when you want the glucose to be consumed, like we can set a lambda value to be very, very less, or alpha G to be very, very high in order to do that. And the second one is Michaelis-Menten's constant for uh, glucose consumption due to glycolic uh, metabolism. Next is the lactate consumption, same. The first one is the cell sensitivity towards the lactate during the oxidative metabolism, and second one is the glucose due to glycolytic. So basically, two lactate has been generated every time when the cell undergoes glycolysis consuming glucose. So that is what this equation is basically explaining. And yeah, so this is how the medium cell proliferation will look like as we see the glucose is consumed. So this is the anaerobic respirations graph. Um, as the glucose consumed, we can see the lactate byproduct is increased in the medium. And once the glucose is fully consumed, the cell stopped proliferating. So the system is now truly anaerobic. And while we observe the system to be truly aerobic, so we have kept the glucose and the lactate. So once it starts consuming lactate, and then later it starts consuming glucose to thrive, and then the cell proliferating here. And the same with the switches. So how XL and XG will work. And we can, so here L star has been set as 10, and we can see like if the lactate is going to be increased more than 10, we can see the switches on, and it is 0 elsewhere. And with glucose, yeah, here the G minimum is set as 0.5. So we can see like until 0.5, we have the switch step function is on, and after that it is off as 0. And both the equations together. So we can see the same pattern. The initially it undergoes glycolytic metabolism, and it started consuming glucose. Once the glucose started to wear off and the lactate is increasing, the medium become more acidic, and the cell will switch from glycolytic to oxidative pathways and starts to consume lactate. So I try to recreate a few figures from the paper which has proposed the mathematical model of the same. So I try to recreate figure 3.3a. So this figure 3a projected the cell proliferation under certain condition for two cell lines. So the blue figure is cell line LN18, and the red lines is cell line LN229. And they have proposed only glycolytic action with the model, and this is the respective output that I'm getting. So initially, so I have set all the initial conditions based on these graphs. So they have started, I assume like they have started somewhere from uh, 0.11. But yeah, so this is the output that I've got. So we have like slight shift in the input, but if you see the parameters marked and their standard deviation with the curve that they have offered, it kind of agrees to what they have provided. And the glucose consumption of for both the cell lines in comparison. So again, it kind of agrees the pattern like uh, the cell line LN228, their glucose consumption is slightly slower because of the chosen rate of glucose and lactate consumption by the cell compared to LN18, which they were trying to explained in the paper. And we can see that pattern, but like for me, I kind of find that glucose consumption was little bit faster with cell line LN18 compared to the paper. But there is no standard deviation specification has been mentioned. So and with respect to lactate concentration consumption with both the cell lines, so same here, like the cell line LN18, they have showed the faster increase in the lactate in the medium compared to cell line LN229, uh, which is obvious because the glucose consumption was faster with cell line 118. So yeah, so this is the replication, and this is the model which additionally I have added with oxygen switch and the oxygen nutrition with the model. So initially what I did, so I tried to introduce the oxygen switch. Here oxygen switch is pretty direct because we are considering only two different metabolisms. One is aerobic and one more is anaerobic, and we should have the oxygen's role play only during the aerobic respiration. So I have included uh, the two switches. Like one is like um, one switch will be when the oxygen concentration is lesser than the threshold. So this threshold, I am setting it same to the lactate threshold. Um, so for example, in one of the paper, the lactate concentration threshold was 10. So I have kept the oxygen threshold as 30 because it should be it should agree to the formula that 1 mole of lactate will consume 3 mole of oxygen. So this threshold should agree to that threshold. So my switch will be on whenever there is going to be the oxygen availability for the aerobic respiration, and rest of the time it is going to be off. And I have created a complement switch for the same, which will help the system to switch back to oxidative. And this is nothing but a complement switch of uh, complement step function of XO. And with the nutrition dynamics, with rate of change of oxygen, so the first equation I have constructed in such a way that um, for oxidate, so oxygen is going to be consumed only by two ways. One is when during aerobic respiration, when it is consuming lactate, and during aerobic respiration when it is going to consume glucose. And the number of O2 molecules which is going to be consumed during lactate is for one lactate is 3, and the number of O2 molecules it is going to consume when during the aerobic respiration of glucose is 6. So that's what this reduction equation says. And this is the model that this is how the model is behaving. So basically, I generated the system with zero glucose and only the lactate. So the cell will choose to be oxidative because there is no glucose to consume, and there is lots of lactate, and the acidity of the medium is so high at the starting, and so they have to take up all the lactate because the cell's preference for the lactate is very high here, and the proportionally the oxygen is being consumed in the system. And yeah, so let's skip on to the exercise. So the first exercise is, how do cells decides to consume oxygen and lactate instead of glucose during aerobic phosphorylation or oxidative phosphorylation? So
Can you zoom in just a little bit? Make it bigger.
I have loaded the model with which undergoes only the oxidative phosphorylation. So we have 10 molecule of glucose and 10 molecule of lactate in a medium. So how did we program, how did we tell the model that it should consume lactate and not glucose?
Well, you can switch your switch thresholds or your rates of conversion to favor the favor the species the type you want.
Yeah, exactly. So here, the threshold that we have that will decide what type of oxidative phosphorylation that it will go is with the lambda. Lambda is nothing but the ratio of MCT1 versus MCT2. So MCT1 says how permeable lactate inside a cell in order to undergo oxidative phosphorylation. And MCT2 is how permeable is the cell for glucose to undergo oxidative phosphorylation. So basically, if I keep lambda as 1, which says like both MCT1 and MCT2 is equal, which means the cell is equally permeable for both lactate and glucose. So if I keep it more, then it will make my system. One second, if I make the lambda more, it will make my system to be more permeable with lactate. So if we see, so I have increased the lambda to say the maximum. So which means MCT1 is higher, which is the cells are more permeable for lactate in order to undergo oxidative phosphorylation compared to glucose. So even though like we can see the rate of lactate consumption is very higher and the glucose is not consumed by the medium at all. So that's the answer. And the second exercise, how does the change in the pH of the medium affect the oxidative and glycolysis metabolism? Of course, this is the point that we have discussed throughout. So once when the lactate is going to be higher because of the byproduct from the glycolytic metabolism, so the cell's medium starts. So it is directly proportional to the lactate concentration in the medium. So that's the answer. And exercise 3, what will happen if glucose and oxygen supply cut down to the medium? So basically, if with the medium parameters that we have set for the tumor cell, as in for the ideal system, if there is not going to be any glucose nor oxygen supply, then the cell will not survive at all, because it has to either have glucose to undergo glycolytic metabolism or oxygen to undergo oxidative phosphorylation. So that's the answer. So this is what happens when the system or our medium lacks glucose or lactate. So basically, the summary is like we have characterized the metabolic interplay between the glucose and lactate. I will switch back to my presentation. So I've explained all these exercises over here, and so these are the key findings with the project that I've done. So it basically says how the system biology approach have worked with the substrate in the medium and the cell proliferation. And yeah, so this model, this work can be in future can be extended with how the necrosis of the cell will take place. So we have studied till how the cell is proliferating. So this can be extended till necrosis, and this can also have include a different pH regulations in the medium. Yeah, so that can make the model more realistic to the tumor microenvironment, which can be result in predicting better metabolic behavior and whatnot. So, so this is the reference paper that I have referred the mathematical model from. Thank you.
Great. Questions, comments? I had one question. So you talk about cell death in the model.
No,
You
I have
mentioned that there's, well, you show curves where the number of cells goes down in time. And you mentioned that in some of your models, the cells die.
Mhm.
How is cell death being represented in the model and the equations?
So right now, I have not tried to uh, represent exactly the cell death. So basically, any equation. So for example, let's take this equation. So here, I have tried to plot uh, the change in the proliferation rate. So very less cell will be proliferating, but this is not a result of cell death, because in order to say that cell is dead, like it has so many different conditions, including uh, space in the medium and all the available nutrition. So that is not being included in this equation. So it shouldn't be, but yeah.
Sorry, could you just zoom in there? What is the y-axis of that plot?
Uh, Cell metabolism in the medium.
No, but what is, no, stay on the curve you're on, please.
Yeah.
What in the right-hand plot with the blue line,
Mhm.
what is being plotted there?
So the rate of change of cell proliferation in the medium.
No, but what does the blue line represent? What is the blue line? If the
Uh,
number is
Number of
0.8,
cells. Yeah.
is the number of cells?
Yeah.
So the number of cells is decreasing in time.
Yeah.
So how are the cells dying?
So I mean, like, if we are counting in that meaning, yes, ideally, this says like the cell number of cells which is undergoing glycolysis metabolism is reducing. But my point here is we did not consume the enough evidence to say that these cells are undergoing necrosis.
Oh, I'm
Be-
not asking about whether the cells are dying necrotically or apoptotically or any other way. I'm
Mhm,
asking mathematically, if you look at the equations that you are working with,
mhm.
how can the number of cells decrease?
So the number of cell which is undergoing glycolytic metabolism is decreased, which means the cell is not undergoing any metabolism.
No, but there only is one kind of cell. If you say that
Yeah.
all the cells are glycolytic, there
Mhm.
is only one kind of cell, and
Yeah.
the number of cells is decreasing.
Yeah. No, the number of cell is not decreasing. The number of cell which is undergoing glycolytic metabolism is decreasing. So, which means the cell started being ideal. But when the cell is not undergoing any metabolism, but this is preferentially not saying like the cell is dying at this rate.
No, but look at your mathematics. What is the equation that generates this curve?
Yeah. So this is the equation which has created that curve.
So what if the cells are only glycolytic? Why is there any switching behavior?
This only the first term governs that equation.
Fine. Perfect. So the first term, look at the first term. Can that first term ever result in a decrease of the population of the cells?
Yeah. When the total population is going to be with the PG, then yeah, there will be no proliferation taking place. I couldn't hear you.
No, but
Yeah.
but
Yeah.
PO is 0. So this always
1 minus PG.
increases. This always increases until PG goes to 1.
Yes.
You can never have PG decrease. But you show a plot where PG goes to 0. I don't understand that.
Since
It's okay. You don't have to think on your feet here, but maybe you want to think about that a little bit. Any other comments or questions? I know you worked hard on.
Are you plotting the like rate of change and that goes to 0? Not necessarily, what is that, P2, PO? And so like the rate of change goes to 0, so there's not more proliferation?
actually, professor is correct. So I know exactly why the model is showing the decreasing function, because when I tried to do the model, I just removed exactly the PO system from it and I kept the system ideally the same. So I did not switch the switches off because I considered like there is not going to be any switches because there is no PO in the system. So these switches are resulting in the decreasing term. So it should not be there. When he asked me, it occurred to me only the first term should be there in the system. So I'm sorry, like I didn't notice that. That's my bad.
That's fine. These are complicated things, so it's easy to overlook something. So don't feel bad that sometimes there are things that you miss. But it's important when we talk about it, it's important to try to work together to catch those things. Okay, great. Dylan, do you want to go? Let me put up a title slide for you and then we'll go ahead. Okay, Dylan, thank you everyone for the presentations. Let's see how things have developed with you, Dylan, in the past few days.
Can you guys see the presentation? Cool. Okay. So my project is dynamic flux balance analysis simulation of dioxic growth in E. coli. So this project was based off of 2 papers. The first one is by Carlson et al. In 2023. A study of dioxic growth experiment using an expanded dynamic flux balance framework. They modeled this dioxic growth process, and then they did some extra stuff, but that was beyond the scope of this. And the stuff that they did that I am following was based on this Mahadevan et al. Paper from 2002, where he introduced the dynamic flux balance analysis process, and he did it in the same process of this dioxic growth in E. coli. That paper was the origin of this static optimization approach, which is the thing I implemented for the dynamics. And then also introduced this regulatory feedback for acetate excretion in the dynamics that just handles some of the like biological realities that cells can't excrete acetate ad infinitum. It's toxic to the cell, so at some point it cuts off, and this is how they handle it in the modeling. And so the key questions that I asked in trying to replicate this, and that they asked, I guess, is can the static optimization approach, assuming steady state at every instant, accurately predict the dynamic behavior of this dioxic shift without knowing the kinetic rate constants, which is the good part about FBA? And then how does the addition of these enzyme constraints, which I'll get into, to the model, and this acetate overflow feedback mechanism improve the model? So just for some background, metabolism, which is what what flux balance analysis is modeling, can be thought of as an economy where like resource allocation is essentially what's being traded. So there's carbon aspects, energy aspects, and then enzyme aspects that all go into this kind of economy. And the goal, at least that we are modeling, is to maximize growth. And so the bacteria have to make these economic decisions in how they allocate all of these different resources in order to maximize growth. And then the biological process that we're modeling is dioxic growth. And so that's a phenomenon where bacteria, if bacteria have multiple sources of carbon available, they sequentially consume them rather than doing it simultaneously. So in this case, glucose and acetate are available, but the E. coli will consume all of the glucose before it begins to consume acetate. And so you can see part of that process is there's kind of different phases in the cell growth based on what carbon resource it's consuming. And then just why this is relevant and why flux balance analysis is kind of used in general is a lot of it is for industrial fermentation. So using these E. coli or other bacteria to produce different products or chemicals that we're interested in, and how can we see what's going on inside of that and model what's happening. And a lot of it is used for different mechanisms of control of knockouts and stuff like that. So being able to model this and understand what's happening leads to like yield improvements and just efficiency of this whole process. So this is a picture of the metabolic network of E. coli. They break it down into these different kind of pathways. So energy metabolism, amino acid metabolism, but really all this is trying to show is that this is an incredibly complex system. There's thousands of reactions, thousands of metabolites. And so this is what flux balance analysis is trying to model. And we'll get into kind of how it does that in a sec here, but this is just to show kind of how complex this problem is. So flux balance analysis is a model that transforms that metabolic network we just saw into a stoichiometric matrix, which records the stoichiometry of the substrates and the reaction or the products of each reaction in that model or network we just saw, and as well as a flux vector, which represents the flow of mass through each reaction. and that's kind of like our stand-in for these unknown reaction rates, which for that metabolic network I just showed, we don't know all of the reaction kinetics that go into it. So this method allows us kind of some handle on how we can model that. And then there's also constraints that are added to the whole thing, and that's kind of been the development of flex balance analysis, is how can we constrain the model and add these constraints more and more to make the mathematical representation closer to the actual biological representation. And so the big, I guess, reason that this works at all is there's an assumption of a quasi-steady state in this process. So the reaction dynamics happen much faster than the translation and transcription dynamics of creating and destroying the enzymes. So we assume that the flux through each reaction is at steady state. So there's not, which just means that there's not an accumulation of any single metabolite in the cell. And this picture in the right here shows kind of what's happening. This stoichiometric matrix has reactions on the top. So each column is a reaction and each row is a metabolite. And then the numbers in our stoichiometric matrix represent the substrate stoichiometry and product stoichiometry of each reaction. And so this is a way we can represent that entire network that we just saw. And this V vector represents the flow through each of the reactions. And in order to actually solve this, we also add a biomass or growth reaction, which we just turn the growth, or all of the metabolites that are required to produce a new cell, we represent as its own reaction with its own stoichiometry. And that allows us to solve this linear optimization problem by just trying to maximize for that growth equation. So some of the augmentations, in which I kind of mentioned, of just adding constraints, one of the ones that we're implementing here, and the most common one, is this enzyme constrained flex balance analysis. That extends the standard model by adding a proteome budget constraint, which just allows us to account for the fact that there's a limited amount of space in the cell, and amino acid and other kind of like resource limitations on how much of any single enzyme and how many total enzymes we can have within the cell. So what this does is it essentially assigns a cost to every reaction. So we model each flux through the reaction based on the turnover rate of that enzyme and the concentration of that enzyme. Rearranging it, we can get the term for the concentration of enzyme. And then we can use all of that information to say that the sum of all of the enzymes, which is the concentration times the molecular weight, has to be less than some total fraction of the protein budget that is devoted to metabolic enzymes. And so what this looks like in practice is in that stoichiometric matrix, we add new reaction columns corresponding to the uptake use or like the production and use of each enzyme. And then we also add these pseudo metabolites representing each enzyme to the metabolites, so the rows. That's one method. The other method is to add a a single stoichiometric term for each enzyme metabolite to each equation that it's representing. So there's kind of different ways that people handle it, but this is one of the more common augmentations that people do to flux balance analysis. The second, sorry, 1 second, let me. Apologies. The second addition is augmentation is dynamical flux balance analysis, which is the crux of that paper I mentioned earlier, which just extends flux balance analysis in time. So it allows us to simulate kind of these different processes throughout time. And the way that it works is we have this initial state of some amount of biomass and some amount of substrates in a medium concentration. And we can calculate the uptake constraints. So this V would be its own individual reaction in our stoichiometric matrix representing the flux of uptake of the different nutrients that are in the medium into the cell. And with that, we can set bounds on each of the equation so that the flux, when we solve it, can only reach the amount that is allowed by our uptake constraints. That allows us, with those bounds, we can solve the FBA in the same way that we normally do, so that quasi-steady state maximized growth rate, which that gives us a growth rate and also a flux of uptake of each of the nutrients in our medium. And at each time step, we can update both of those values so that we can get essentially our new biomass and how much of each of the nutrients in the medium were either secreted by the cell or uptaken by the cell, which allows us to dynamically get these values that we're tracking. So this whole thing is called static optimization approach, which was the approach developed by Mahadevan in that paper I mentioned. And so we just iterate this entire process and then that allows us to like track these different values through time, kind of similar to how we've done in Tellurium. So kind of the math of it, which is this is similar to what was in the last cell, but we have at each time step this inner problem where we solve the flux balance simulation. That gives us fluxes of uptake and a growth rate of the cells. Using that, we can calculate the change in substrate concentration and the change in biomass, and then iterating, we can get to simulate this kind of dynamical process. So the experimental data for this diauxic growth that I'm modeling came from that Carlson paper that I mentioned. It comes from a actual batch fermentation experiment in a certain medium, M9 minimal medium, which has things like glucose and certain like metals that are vital to cell growth. And you can see that we have this diauxic shift process here where glucose initially is consumed. Acetate is slowly excreted into the extracellular medium. And then throughout all of that, the biomass continues to grow until it runs out of carbon substrate and slowly begins to decline. And something to just notice and keep in mind throughout all of these is on this right axis, we have a different scale because this is measuring the biomass, whereas in, what is that, grams per liter? Whereas on the left axis here, we're measuring the concentration. So that is going to relate to the glucose and acetate in the extracellular medium. These are the different parameters that I added to the model to have control of. This max acetate excretion and acetate saturation are how I control the acetate feedback mechanism that I mentioned earlier. So this just gives us kind of a bound on how much acetate we allow in the extracellular medium. And this max acetate excretion gives us a bound on the flux of acetate outside of the cell at any time point. This enzyme pool cap is related to the enzyme constrained model, and this is just telling us that only 9.5% of the total proteins in the cell is devoted towards metabolic enzymes. So this came from literature, and I just kept constant throughout, but this is just showing that only a certain fraction of the total proteins are devoted to this. And then one of the other controllable parameters that can be controlled in like the bioreactor that the experiment was measuring is oxygen uptake. So this corresponds to the oxygen, essentially concentration in the fermenter. So this just allows us a way to control how much oxygen the cell can uptake, which controls the difference between these different ways that carbon utilizes glucose. I forgot to go into that, but there's fermentation, which the E. coli uses when there's not enough oxygen, or when there's not enough proteins to do enzyme proteins to actually run the more complex respiratory processes. So when oxygen is available, bacteria can respirate, which is much more efficient in how much energy we get out of each glucose molecule, but it uses heavy and very costly enzymes. So there's kind of this trade-off that we're going to see in some of the simulations. And then just the time step, which allows us to control kind of that dynamic inner and outer process. So these are the results of the simulation that I ran. This is showing us I ran it under 4 different conditions. One of them is standard flex balance analysis without any enzyme constraints and without that acetate feedback mechanisms. So in this case, the cell is allowed to excrete as much acetate as it solves for optimal growth. And there's no limit on how much flux can go through any specific reaction, which is really what that enzyme constraint is controlling for. In this top right here, we see the same standard flex balance analysis with that acetate feedback mechanism. And you'll notice that we lose this spike here, which represents our simulated acetate. And we see it follows much closer to the simulated data where we don't see the spike and there's only a certain amount of acetate that can be excreted. And then bottom left, we have the enzyme constrained version without acetate feedback, where we see the same process of this acetate spike, which is biologically infeasible. And then finally, all of the constraints added, so enzyme constraints and this acetate feedback constraint. And you can see that we get much closer to the actual experimental data. I don't know if I don't have a slide for it, but I will say this is using the values for all those parameters I mentioned from the Carlson paper. So right away, we can see that this doesn't match the experimental data in the same way that their paper did, which I will get into kind of reasons for why that might be a little later. Following that kind of initial analysis, I did sensitivity tests on those three controllable parameters. The first one is just this O2 sensitivity. This was something they mentioned in the paper that was also something they controlled for and did something similar. So we can see that at low levels of O2, we get much slower uptake or much slower like usage of glucose, slower excretion of acetate, and essentially minimal growth here, which is to be expected because this drives E. coli into that less efficient fermentation mode. So there's not as much energy available for the cell to grow. But as soon as we start getting up into above 10, we see that we get much closer to the experimental values. And this parameter gives us a way to kind of control the levels of fermentation versus respiration that the cell's doing. And through this, I found that the optimal value for our simulation, or closest to data at least, was 10 millimoles per grams dry weight per hour. The second sensitivity analysis I combined the Vmax for the acetate feedback and the kinetic parameter for the acetate feedback, and just plotted the best results we got here. So this is we can see gets us much closer to kind of experimental data, and the kind of most important parameter we can see here is this. These are all the top 5 best results, but the most important parameter is this Vmax at 8, and then just a high K value we found to be the best results. This is our results using those optimal values parameter values that we got from that sensitivity analysis. So on the left is our results where we used O2 of 10 millimoles per gram dry weight per hour, the Vmax parameter of 5 millimoles per gram dry weight per hour, and then this kinetic constant of 50 millimolar. And that's in comparison to this figure is from the Carlson paper, where they match the data much closer. All their solid lines are their simulated data. But these are the values that they found, 10, 2, and 13. And so the reason that there's kind of this discrepancy in between what I did and what they did is I had a very stringent essentially like leak proofing of my cell of all the excretion of like not glucose or acetate. I essentially just set to 0, which is biologically infeasible, but I was getting issues with it was just uptaking whatever like carbon sources it wanted at any value. So in their model, they must have some sort of much more precise and probably realistic amounts of extracellular concentrations and flux values of these extra different metabolites that are produced by the cell, either uptaken or excreted, and not including that in my model, just being too stringent on what I allow to be move in and out of the cell. This is the best that I could get with the simulation data, but the acetate feedback was pretty close. So now I can go into I did 2 separate notebooks here, one which is really in depth and has all the code, and then this kind of simpler one that just has the exercises and a simpler kind of background and all the things that are going on. So going through the kind of more educational one, this we're modeling this dioxide growth process, which I mentioned before, where glucose is consumed all the way before any other carbon source is used. And what we see in that model that I was going into is there's 3 different kind of processes that are happening that we're trying to model. The first one is respiration, which occurs when there's a lot of oxygen in the cell. This process has a very high yield of ATP. And it yields 10 times more ATP per glucose than fermentation. Fermentation is the process the cell uses when there's either oxygen is limited or the respiration enzymes proteome budget becomes saturated. So if the oxygen level becomes too high, then fermentation will kick in so that the glucose or the cell can utilize as much glucose as possible at the expense of efficiency. And this fermentation process is actually where we see the overflow metabolism of acetate being excreted into the cell, because acetate is a unwanted kind of result of this process. It's toxic to the cell, and once it's reused, it's not quite as efficient. And then the third one is acetate. So once all the glucose is used up, the cell can use acetate as a carbon source and fuel source. So it converts the acetate back into acetyl-CoA through the TCA cycle. This has a higher ATP yield than the fermentation, but lower than glucose. So we still see cell growth here, but not as high as we see in glucose. This is a little more in depth of the math that's going on in that flux balance analysis I mentioned in the slides. So you have your stoichiometry matrix, your flux vector. You set it equal to 0, which represents that quasi-steady state assumption. And then each reaction has a lower bound and an upper bound of which controls like which direction that each flux or reaction can go. The enzyme constraints here, which we went into a little bit, and then the math of the dynamic process. So in order to actually simulate the dynamic process of biomass growth, once we solve the flux balance analysis, we update the concentration or whatever amount of biomass based on the previous time step biomass times this e to the ut, which is the growth rate, which is the final output of that flux balance analysis, the thing we're trying to solve for of an optimal growth rate, and then our time step. And this allows us to get a time series of how our biomass is growing. And then we do a similar process for the glucose and acetate concentrations. So the concentration at the current time step is based on the concentration at the previous time step, plus the flux that was solved for in the flux balance analysis, times the biomass at the previous time step, and then times our time step. So all of these things allow us to kind of plot the dynamics of how these different biomass and then concentrations are changing. And then this is how I modeled the acetate feedback, which came into play in not in the flux balance analysis part, but in the kind of extracellular. I guess it does come into play in the flux. So it's essentially an additional constraint that just sets the upper bound of the acetate excretion, or so that based on this like logarithmic equation here, this was just gotten from the Carlson paper and implemented as they implemented it. But this just gives like logarithmic negative feedback based on how much acetate is in the extracellular medium. And then, that other page just has more detailed explanation of all this stuff. So For the exercises, before I do this, we're going to go a quick one. So this model is showing us, or this picture is showing us the different model conditions, flex balance analysis with acetate feedback, without acetate feedback, and then enzyme constrained with and without acetate feedback. So first question is, which model will produce more biomass, standard flex balance analysis or the enzyme constrained flex balance analysis, and why?
Can you show the question again?
Yeah. So the standard flex balance analysis is just the generic reaction stoichiometry matrix, and the enzyme constrained one is where we add those extra equations that limit that add extra constraints to how fast each reaction can go. Yeah. These are the same ones we did in class. Do we want to go through all these?
If you did the ones you did last time, you might do one more,
Okay.
but I think since we covered, if they're the same ones we did last week and you haven't changed it, we don't have to do all of them again.
Okay. There might be a couple at the end, but I didn't change any of them. I just don't know if we went through them last time.
Maybe jump to one of the later ones so that we can see one of the ones we didn't do last week.
Okay. They're based off of this one too, so I'll just go through. This is that same kind of one I showed in the slide, but this is with acetate feedback. You can notice that spike goes away. The enzyme constrained version versus the standard version. Enzyme constraints just slows down the whole process. So this way you can kind of see how each of these different constraints affect the results of our simulation. Went through that one. This is showing us that sensitivity analysis essentially of different values of O2 and how it affects the simulation. So we can see here that when the available O2 is really low, we get essentially no growth because all of the glucose consumption is essentially driven to fermentation. So since that's so inefficient, we don't see much cell growth. As we increase the amount of available O2, we can see that the cell growth increases, which is just kind of showing this process of the switch from all fermentation to some balance of using respiration now, which is much more efficient for the cell so it can grow better. I think we went through all the only yeah, so this one is kind of longer, but we can go through it. So for each feature, so if we get rid of the enzyme constraint, what effect does that have in the model behavior?
Could speed up the growth, I would have thought.
Yeah, well, it speeds up the growth and the consumption. Essentially, the whole process gets sped up much faster because because our model can just flood any reaction with as much flux as possible. Okay. What happens if we get rid of the acetate feedback mechanism?
Well, you should accumulate a lot of acetate then.
Yeah. Yeah. So we can see here that just is a limit on kind of the acetate that can be excreted into the cell. So when we get rid of that, we see this huge spike here, which is just biologically infeasible because you get some sort of toxicity much lower than that. So that constraint was actually found to be like the most kind of important one for actually matching real world data. And then last one, if we decrease the O2 level, we kind of went through going through the slides, but you can see that the cell is forced into fermentation. So since that's such an inefficient way of producing or of, I guess, generating energy, that we see a really small amount of cell growth. I think I don't know, it's at least 10 times worse than respiration. So it's much less efficient at generating ATP. Yeah, and then I guess just kind of closing thoughts. I mentioned that I didn't quite match the data in the way that the Carlson et al. Paper did. And the reason for that was just kind of an expediency and like an unfamiliarity with the process of essentially shutting the cell off to every excretion uptake of anything except for glucose and acetate, which we were interested in. So that kind of limits the amount of like actual biological feasibility and like products that can be made within the cell. So that will limit the growth that can be achieved and the actual like enzyme, really everything, but the like enzyme constraints of what's made, what's not made, will all be affected by that. That being said, though, this even without like matching the data perfectly, both of the constraints that we added matched the actual experimental data much closer, which we could see when we kind of compared those two things. So the enzyme constraints had the effect of like slowing down growth, which was much more feasible, and then the um, acetate feedback just limited that acetate spike and kept it much closer to feasible ranges. So like the takeaway is that this whole dynamic process works for modeling all of these different behaviors, but in order for it to be useful, you have to actually add a fair amount of constraints that are um, based on, I don't know, having some like background knowledge of the actual biology. So as that builds up, there's been much more impressive models based on flex balance analysis. And that's what I got.
Any comments or questions about that? You've added a lot of material since last week, especially to the introductory part.
Yeah, a lot of it was just cleaning up kind of I don't know, making it clear of some of the background knowledge and going into the math and all that stuff a little deeper.
Anybody else? Sorry, go ahead.
Just saying thanks.
Okay, do people know? Sorry, go ahead.
I was gonna say, if there's no comments, do you mind if we go through? I did this, but I'm not sure if this is what you're looking for. I was wondering if we could just finish class off with.
Oh, that was exactly what I was going to say. If we have time, I'd like to do that. So you went to Zenodo and you put up this is what I was calling the landing page, where you have a description, a brief description of what the purpose of the model is. It's probably important in your cover to say that this is a primarily replication of some papers. So you want to make sure you cite the source papers in that cover thing.
I mentioned them, but then go into the full reference down here under reference.
Right, but yeah, okay, so that's fine. But I think that looks fine. Okay. One problem with Zenodo, of course, is that once you published it, you can't change it very easily. Um,
I did notice too that it didn't allow me to keep my file structure, so it just made everything its own file in the same thing. So some of the paths might get a little messed up that I didn't catch until it was already published.
Pedro, do you have any suggestions on that?
I mean, if he done it, he done it.
I also have
There's
a GitHub page which has the file structure and everything. I put that on here also, so that might be a
Right.
better way to go
Right.
about.
So if they want, they can go to the GitHub and then they preserve everything. That makes sense.
yeah. So whatever is in Zenodo refers to the version of your GitHub files at the time you submitted to Zenodo. Now, if you were giving the repository URL, then it's fine, but the user has to skip all of that and go straight to your GitHub to see what's new. So
Yeah.
it's basically giving a DOI to a version of your GitHub repository,
Yeah.
which is limiting, but it's fine.
Is there a way to like, I guess it might be too late if I can't change it, but like add file structure?
Well, it'll give you whatever you do, it'll give you a new DOI.
Yeah.
Because the whole point about a DOI is that once a DOI is issued, you can't change anything.
Yeah.
And it's okay. I mean, you can have a dead DOI that represents an earlier version, but you don't want to generate too many of those. In other words, it's
Is it
okay to have
possible
a
to
version
delete
1
one?
and a version 2, but you don't want to have version 1 through 27.
Okay.
Go ahead, Pedro.
Maybe you could delete one. I don't know.
I don't think you're allowed to. I think once you have it, that you shouldn't be allowed to delete it. Once you have a DOI, you're not supposed to be able to get rid of it.
Yeah, no, you cannot get rid of it.
Meh. You can delete the
Oh.
record. I don't know if that deletes the
No.
DOI.
No, once you click the publish button, it's done.
Yeah.
You cannot undo it anymore.
Um, is this okay, then is this what I turn in? I guess is a link to this.
Well, that should be included with what you turn in, yes.
Okay.
Everyone needs to publish the documents. And I was talking to Hayden and Pedro today. You may be tired of this, and what you submit today is fine. But if there are any last tweaks you want to do before the final submission, you have until 5 p.m. on Thursday. And so whatever you have uploaded by 5 p.m. on Thursday will be the basis for the final grade for the notebook. Clearly, you can edit your slides too, but of course your presentation was the presentation you gave today.
Okay.
If you want to put up exactly what you have today, that's fine. But if you noticed while you were doing it, oh, I need to change this or fix this, you have till 5 p.m. Thursday. Sometimes giving people more time isn't doing them a favor, but I want to make sure that if you feel you need it, you've got it. Anything else on the Zenodo side of things that you want to go over? Lakshmi or Roying, do you want to discuss that at all?
I just wanted to ask, like, everybody needs to publish our report here, right?
Yes, that's right.
Okay.
That's required. I can't grade you on the class unless you have a Zenodo DOI. because your code is fundamentally not distributable until you do that. Okay, well, I want to thank everybody for working hard this semester. I've enjoyed working with you. I look forward to the final versions. And I appreciate all the effort and learning people did, and especially your patience in presenting and then reviewing and then modifying and presenting again. I know that that can seem like a lot, but it's part of the process of doing scientific or technical development, is to go over the same thing multiple times and do refinement. And so I do appreciate your patience with that. And happy to talk to you about other things or the class going forward. I'd be happy to see you in class next semester if you want to take that. And I also want to wish you, if I don't talk to you before the end of the year, a great holiday and a great new year. Thank you, everybody, for all the hard work.
Thank you, Professor.
Thank you, guys. Thank you, Pedro and Nathan, for I don't know,
No
all the
problem.
help. And that was one of my favorite classes I've taken. So
All
big
right.
fan.
Are you
Well,
taking next semester?
No, will it be offered next year? I'm only taking one next. I have to finish out my minor.
probably not, but you never know.
Yeah. No, I didn't get much research done this semester, so I'm trying to focus on that next semester.
I hear you. It's hard to take a lot of classes and do research as well.
Yeah, but no, I appreciate it. Maybe I'll drop in or look at the slides on YouTube.
You're welcome in any form. If we want to talk more about modeling as you go forward with your own research, that's fine too, whether you're in a class or not.
Yeah, yeah. I mean, I'd love to. I don't know if I ever do any dynamical stuff. Just get in touch, get your expertise.
We
Yeah,
also
just reach
teach
out.
that the spring class is also offered over the summer as a free uh, week to 10-day long workshop. So
Oh yeah, I could do that.
that maybe takes less it's more intensive, but it takes less time, less of an extended period of time.
Yeah,
And
that actually
everyone,
sounds really nice.
and
It's a
that's
boot camp.
available to everybody. That's available to everybody anywhere in the world for free.
It's like a boot camp. It's probably one of the most fun things we do, and people usually love it. They have a great experience working together. They work in teams, building models and simulations of a certain biological systems they're interested in, and it is extremely intense, but it's fun as well. So I highly recommend it if you're interested in continuing with modeling and you cannot attend next class. That would be my recommendation for you.
And that one is it's all online, but one big difference is in that case, we typically have between 25 and 50 people taking it. And so some of the things that maybe didn't work as well this class, where we were trying to do things uh, in a collective way, when you have 35, 40 people, you have plenty of people to build teams and review together and work together and support each other. So some of the structure that maybe doesn't make as much sense when you have 3 people in a class makes a lot of sense when you have 50 people. Hayden is one of the lead organizers, and Pedro does a lot of the teaching. So the
Is there
team
a
is the team.
is there like a way I'd like more information resources on it? Is there like a mailing list or something of a reminder so that I don't forget after a semester?
Yeah, I'll make sure you're on it.
Okay.
Announcements usually start going out around March.
Okay. Cool.
But yeah, we'll add you to the mailing list. But if you have any questions or want to know more about it in the meantime, please feel free to let me know.
Okay.
I mean, it's all online and on YouTube, but getting through it on your own is possible. It's designed to be possible, but it's also intimidating in the sense that if you don't really have the sort of carve out the time to concentrate on it, getting through all of that material by yourself can be heavy. So it's more fun to do it as a group.
Yeah. Cool.
All right. Well, thank you again, everybody. Thank you, Hayden. Thank you, Pedro, for all your hard work as well. And we'll look forward to what people upload in the next day or two. And again, best wishes for the holidays and the new year. Good night.
Same. Good
Night.
night.
Bye, guys.
Bye.